And I will discuss the clinical trial. Some will be as repetitions of the lectures that I have previously explained it, and some it will be anew but in a good view. So the first one is the aim of a clinical trial. So we discuss what are the aims and what are the goals of a clinical trial. So if I discover a new drug or a new device or a new diagnostic treatment or anything that I can do it, we just do something which is called a clinical trial, and it's not easy at all. So the aim is development of a new medicinal products and medical devices, or improvement of the medical treatment. Maybe there is a drug that is already present, but we need improvement of this medical treatment. It may be a new diagnostic method, it may be a new treatment option. So this is, or these are, we can say, the aims and the goals of a clinical trial. So we have here the map is the clinical trial outcome. Okay, I do already, or I have did the I have done the clinical trial, and we wanted to know if we have this clinical trial outcome. So we have several questions around it. We have significance, relevance, internal validity, and external validity. And we wanted to know what are the differences. So the first one is significance. Significance, is the result significant? Is that is important? How it's important these results? That's mean exactly the significance. Are the treatment arms, groups of different, I don't know, but it's the result significance. It's it has a great effect or not. And the relevance, how meaningful is the data? I mean, how it's relevant to our goal, how it's affected us, how it's help us to achieve our goal. This is the relevance. And there is this internal validity and external validity. Internal validity, how good the measurement tool. It's about um, the tools that we use it, the measurement, the analysis that we use it, how it's accurate, okay? That's its internal validity. So I think internal validity, it's about how accuracy these results. And about the external validity, does the trial design allow a translation to the general population? So when we just do the clinical trial, we just, choose a group of the patients or a group of the humans, or, but for sure it not represented the whole population all over the world, but we try as we can that it's represent the whole population and how it is affect the whole population. So again, internal validity, it's about the accuracy. External validity, it's about generalizability. And the significance, the importance of the result, the relevance, the relevant of these results. And these are the discussions of the clinical trial outcome. So we put two sides, data level, trial design level, because the trial design also, it has many design, and how that is affect on the patient population. So we have a multiple types of these clinical studies. So the first one, it's interventional study, and the interventional study, it's completely the clinical trial. And I wanted to talk what is the difference here. Interventional study, and the word interventional, that's mean I do effect on this person. There is something, it's not just only observe things. No, we have this effect on this person. Okay, let's do it. Treatment is a part of a study, doesn't reflect daily clinical practice, like blood sample, drugs, making subject to something like intervent. It's like I mean, I do something with these patients. I mean, I ordered them to do something. That's it. I just not observe it from away and they don't know anything. No, that is exactly what is observational study. The other thing, it's non-interventional. But the interventional study, um, I do the treatment that is a part of the study, but do not reflect the daily clinical practice. It's not associated with the clinical practice. I mean, it's not reflect the daily clinical practice, but I do the treatment, I do a thing to these patients or to these person that I have chosen them. So like, for example, blood sample, because I take a blood from them, drugs, I do treatment on them, making subject to something, I do some effect on this person. That's exactly what the intervention. Why I didn't understand it is that is not reflect the daily clinical practice. Is something it's not exactly what is happening in a daily clinical practice. But observational study, I mean here it's just observe this person, observe this group that I have choose them, maybe know about me, maybe no don't know about me, but I do not do any effect to them. And that's exactly why I said non-interventional. And we have other types of studies also, like retrospective studies and prospective studies. And we say retro, it's a Latin, and that's mean it's behind or something, it's backwards. So retrospective study is a study of the events that already occurred. So that's exactly what the retrospective said. I study the events or study whatever I study it, but it's already occurred. So if it's I study something that already occurred, so don't have any effect on this, and that is exactly observational. We can say that is observational. Okay, so we have a retrospective studies. We can say observational because it's a study of something that already occurred. But a prospective study, I study things that is happen in the future, like following this condition, following these patients. Maybe I have their effect, maybe I do some effect on them, and then we can say interventional study, or just observe them, and we can say observational study. Okay, good. Now we will discuss something so important, like general map of these things. So we have clinical studies. In the clinical study, they can maybe subdivided into two things: observational or non-interventional, and there is something is called non-observational interventional. So let's focus on observational or non-interventional, not interventional, like cohort study, like case control study, and others. Cohort study, like I make a group of the patients or a group of the person and I follow them. Case control study, like subdivided these persons into two types, some two groups, some they are case and some they are control, and others. But the cohort study, case control studies, and others, they are just a part of the observational or non-interventional studies. But when we're talking about the interventional studies, which we can say in practice they are the clinical trials, because the clinical trials are interventional studies, and they are subdivided into two types: controlled or non-controlled. If they are controlled, then we have three types of a study. We can say parallel study, we can say crossover study, and we can say it's sequential study. And we will discuss all these later, but we just wanted to understand what is the difference of the interventional and non-interventional studies, and we just focus that the cohort study, case control study, because we will study it more, it's under the observational study, where the crossover it's under the interventional studies. So we have a control, and the control that is subdivided into parallel, sequential, and crossover. And then we discuss the phases of a clinical trial, because we will repeat it again and again and again. These are, it started with a research, then the preclinical, then the phase 1, phase 2, phase 3, then registration, then phase 4. So the first one is a research, and it's about the target and the molecule. That's we discuss what is the target. The target, it is can maybe enzyme, it can maybe protein, it can maybe receptor. We know that the enzymes are a protein, but it's have several things, and the molecule. I mean here the target and the molecule. Molecule is the drug, maybe, for example, and the target, that's mean the drug, it has effect on the enzyme or the receptor or something. So that is a research. We have to identify the target, head, leads, and these things, and we will discuss it later. And the molecule, which is a drug itself. And then the other phase is preclinical, and the preclinical is on the animals. We do it in the animals. So we study the toxicology, pharmacology in the animals, galenic production. I mean here by the galenic, that's mean we understand how it's the absorption effects or something. But in general, the preclinical, it will be experiences on the animals, and we discuss all the toxicology and pharmacology, but it's in the animals. Then we start all these phases, and these are in the humans. Phase 1 first in humans, and that is maybe healthy individuals, but phase 2 it's in the patient, and phase 3 it's a proof of efficacy and safety, how it works well, how it's safe to the patient. So phase 1 it's first in a human but in a healthy one, and phase 2 in the patient. Phase 3 we have to make a proof of the efficacy and the safety. And then we go to the registration and approval, like we check up by the FDA and other organizations. And phase 4 it's in the marketing level, evaluation in the real clinical settings, that we can do the post, we can say it's a post marketing surveillance. We just send it to the real population and watch out what is happening here. So we can say the research and the preclinical is under the preclinical development. Phase 1, phase 2, phase 3 it's a clinical development, registration. Phase 4 it's a post approval. And the other thing I will discuss about it is the cost per launch. What is the cost of this? It maybe reaches to the $1 billion, and that is according to the USA, and the time it's 12 years. It's maybe from 10 to the 15 years. So it has cost, time, risky, multiple things that we will discuss it later, but we have just a view of that. There are two persons we will discuss about them: sponsor, and the sponsor is the person that is responsible for writing of study protocol. It may be not one person, it may be organization. Okay, so writing the study protocol, which is an important thing, organization of study database, drug investigator, and data analysis, writing of a study report. So we can say that the sponsor is responsible for writing a study protocol and writing of a study report, which is so important, and sponsor responsible for the investigator, the drug, the database, the everything about it. And it's the person that is responsible the data analysis. And again, I said the sponsor, maybe it's not the person, maybe it's a group of people, they act as a sponsor, but it's not usually it's a person. So but it's important to know what are the responsibilities of the sponsors: writing a study protocol, writing a study report. It's also important for the data analysis. It's responsible also for the investigator. About the investigator, it's a physician or a dentist, and the treatment of the patient according to the study protocol, collecting the data, the source of the data, transfer of the data, sponsor. So now the investigator, we know exactly what is investigator. Investigator, it may be a physician or a dentist, so it's a medical person, and is responsible for the treatment of the patients according to the protocol. He should follow the protocol and collecting the data and put the source of the data and send it back to the sponsor, because the sponsor is responsible for data analysis, for collecting these data. No, collecting the data, it's the responsibility of the investigator. Okay, so again, discovery, we can say research, preclinical, and the phases, these clinical phases. So again, phase 1 it's in the healthy individual. We study actually the human pharmacology. We determine the safety, the dosage, and the pharmacokinetics. So this is the phase 1. We determine the safety. We determine just the first step in the safety. Okay, the dosage, like we should know what is the maximum dosage on these things. Pharmacokinetics, we know now pharmacokinetics, it's about ladma, liberation, absorption, distribution, metabolism, and elimination. So we study all these things: the human pharmacology, safety, dosage, pharmacokinetics. But phase 2, because it's present in a human body, so we can say it's a therapeutic exploratory. And we say exploratory, that's mean we are doing now this on the patient, and maybe we discover something that we don't know before. So evaluate the effectiveness, how it's effect, and maybe we discover something new while we treated the patient, because this is the first time that we treat the patient, not the healthy individual. And then the phase 3 is therapeutic confirmatory. That's mean we confirm the effectiveness, we confirm the safety, we confirm everything. So we can say phase 1 is a human pharmacology, because we study the safety, we study the dosage, we study the pharmacokinetics. It's the first part that we can do in a healthy individual. And the phase 2 we can say exploratory therapeutic, because it's the first time on the patient. And the phase 3 it's we confirmatory, or we can say therapeutic confirmatory, because we confirm the effectiveness and the safety. The questions is the number of the subjects. In a phase 1 we want 10 to the 100 volunteers, and most of the time it's not more than 100. Phase 2 100 to the 500 patients, not healthy individual patients. And phase 3 we do that to the 1000 to the 30,000 patients. See, that's a huge number, just to ensure everything right. So phase 1 100 to the 10 to the 100 volunteers. Phase 2 is 100 to the 500 patients. Phase 3 is 1000 to the 30,000 patients. And we can say phase 4 because it's post-marketing testing, or we can say it's a post-marketing surveillance, because we do that to the whole population. So it will be more than 10,000 patients and more. Threats to the internal validity. We discussed before what is the internal validity. That's mean how is the measurement tool accurate, how is correct. But there are many threats, many problems that this can maybe happen. Bias, confounding factor, random error. But we will discuss what each concept has a many definition, and we have to know it. So we have many threats to the internal validity. So the first one is bias, the second one is confounding factor, and the other one is random error. So what is a bias? Bias actually is the systematic error that leads to the inaccurate estimate of an association. So the first thing that I wanted to focus, systematic. It's not local, it's not it's systematic, and that's so important to focus on this. Okay, so that is related to the systematic error that leads to the inaccurate estimate of an association, because I make an association between two things, maybe for exposure and the disease or something, but there is something wrong that I do it, and that is lead to the bias. Non-random systematic error in design, recruitment, data collection, analysis. So we have many types. Actually, when I say non-random, because the errors subdivided into two types, systematic and random. So if it's systematic, that means it's not random. Okay, so we have two types of the errors, systematic and non-random. And bias is systematic error. Again, I said bias is the systematic errors, but these errors that is maybe contains in a design, like when I say crossover, case control, code, or something. It may be the bias in recruitment, it may be the bias in the data collection, it may be the bias in the analysis of the data. So we have many types. The first one is selection bias. Selection bias, systematic differences between the baseline characters of a groups. That's mean I just made many types of the groups, and each group they do not have the same individual, and except that I do that the results it's for the individuals. I just make group 1, group 2, group 3, and each group that is contains individuals, but they are different from each other. There is nothing like similar concepts or similar characters, and that is lead to selection bias. Performance bias, different additional care between the groups. The selection bias is, when I choose these individuals, I was not be careful about the characters of this individual, and that is maybe affect the results from these groups. But performance bias, like I deal with this group or I do something with this group that is different from the other group, and despite all these things the results for all the same, and that is lead to the performance bias because I do something or additional care or special deal with this group, and that is lead to the performance bias. Observational bias, different reporting of the outcomes, and we can say it's observational bias because I do a report of the outcomes, but it's different reporting. Maybe they are the same, but I do some positive here more than here. These are what we can say observational bias. We have another types of the biases like recall bias and deduction bias. So we have many types of the bias. So we have selection bias, performance bias, observational bias, recall bias, deduction bias. So we know that the bias, and that's important for me, as a systematic error. It's not random at all. Now I discuss what is the confounding factor, which is another threat to the internal validity. It associated with the expose but not consequence of exposure, associated with the outcome independently of exposure. And we know that by the examples like smoking, coffee drinking, and heart disease. Okay, like this example. Okay, now we have to understand what that means. It associated with expose but not consequence of exposures. Like it's not something that is related to the expose, but it's not consequence of exposure, and it's associated with the outcome, but it's not related to the exposure, like the cases of the Down syndrome by maternal age. How that is happen? Like maybe the outcome or the third factor, and I think it's more easy when we see that is a coffee drinking that is related to heart disease. Coffee is exposure, outcome is a heart disease. And there is something is called smoking, because who drink the coffee maybe is smoking. It's somehow that is related to the exposure, but it's not a consequence of it. And smoking at the same time is associated with the outcome. It causes heart disease, but it's not related to the exposure. And that's exactly what I mean exactly by the confounding factor. It's a third variable that is associated with exposure, associated with the outcome, but it's not like a sequence of that. Random error, which is the second types of the error, decreases with the study size and leads to imprecise results. Sure, if we just do the random error or systematic errors, both of them that is lead to the inaccurate or imprecise errors, but want you to focus more about the words that we choose it here. The other thing is random error that leads to imprecise results, and systematic errors lead to the inaccurate results. What is the difference here? We can say he said that the random errors decreases with the study size and constant despite increasing the study size. Whatever the systematic errors, like a bias that we discussed at, that is absolutely lead to something is called inaccurate results. The results are false. We can't use them. We can't focus on them. We can't, confirm it because completely false. But when I said random errors, imprecise results, that lead to results, but they are not. We can say it's accurate, but they are not precise. They are not details. Maybe we can say okay, we can do with these results, we can deal with these results, but they are imprecise. Okay, so that is the difference. And the other difference, if we increases the number of the study sites, I mean, I increasing the number of a population and a persons, that it will be decreases, and maybe I don't see any errors again. Okay, by increases the study size. Okay, because you see decreases the study size, I mean the errors decreases, the random error decreases with the study size. So if I increase the number of the person, then the error it will be decreases. So that is lead to the precise results, and I will be confident about these results. But we use it anyway. You know, distinguish. But about the systematic errors, they are constant despite increasing the study site. Whatever I do, the population, whatever I increasing the number of the persons, whatever I do, error is error. The results are inaccurate, and that is more, I can say, horrible, defying things, because the results are inaccurate anyway. So these are the difference between the two types of the errors. So we have random errors, systematic errors. The random errors decreases with the sample size, and that is lead to the imprecise results. But the systematic errors constant despite increasing the study sites, and that is lead to inaccurate results. Now about the analysis of the clinical trial results, significance of the differences, medical relevancy of different studies, control of challenges to the internal validity, external validity, similarities between the subjects and overall population. And I discussed that before. I said significance, relevance, internal validity, external validity, and I explained all of them before. We have another thing, which is what is the purposes of a clinical trial. And I said before that is a new treatment, a new diagnostic therapy, new something, but we will discuss is more details. So we have treatment, like the first thing that we do, treatment of the disease or the syndrome or the condition. This is the purposes of the clinical trials, and this is the first goal, is a treatment. Maybe we wanted to treatment of disease, syndrome, and condition. Is there a difference between disease, syndrome, or condition? Yeah, maybe it's condition, that's maybe the patient does not suffer, have the symptoms, but there is no suffering. And syndrome, it's a we can say a group of the symptoms but with no cause, and the disease we know already the cause but we wanted to treat it. Or a prevention, like a prevention development of a specific disease or health condition. It may be diagnostic identification of a disease or health condition, or it may be supportive care, like maximizing of comfort, minimizing the side effects, screening sometimes, and that is important for the cancer diseases, like identifying the condition or risk factors for a condition. It may be important for the health services research, like evaluating processes of healthcare, health services research. That's we do evaluation, and we just observe the services of the healthcare and we wanted to improve it, like this. Basic science, examining the basic mechanism of the action. Maybe there is something that is can maybe happen, or a drug or a disease, and we wanted to know what is the mechanism of the action, so we can say it's a basic science. And the other thing is device visibility, device product evaluated for feasibility of the product. Other, like none of the other options applies. So we want to summarize it again: treatment, prevention, diagnostic, supportive care, screening, health service research, basic science, device visibility, and others. So primary purpose or research hypothesis, study type we have to do if that is observational, interventional, retrospective, prospective, and the trial design. So we have allocation, randomization. That's exactly we want to discuss these things, exactly what each one of them know. So if it how it's the allocation or randomized, if it's randomized or not, and we will discuss it. Intervention, model, and control, like what is the arm type, what is the number of the arms. We will discuss also the masking and the blinding, the type of the masking and the blinding, open label, single blind, double dummy. And we have the outcome measures, we can say endpoints, we can say primary, secondary, exploratory, and endpoints. And the study population who is enrolled, like what are the type of the population that we will involved it in this group. And that will be the next lecture. We will discuss all of them in more details. Thank you so much for listening. Hope you understand anything.